Hello everyone, welcome back to my video tutorial for data integration in Surat. In my previous video for data integration using Harmony, we loaded 3 single-cell RNA sequencing dataset for healthy human lung, and we performed the standard workflow analysis, and we showed a batch effect among 3 samples. You can see here we named the batch samples as AML1, AML2 and AML3. For the standard workflow analysis, the samples were not integrated very well from different batches. I already showed you in the previous video we can use the Harmony function to integrate the data. Today, I'm going to show you 2 other methods, FastMNN and another method, the Liger function, to integrate your dataset. So we already performed the data loading, quality control. If you want to use these 3 datasets for your practice, you can watch my previous video using Harmony to load the data and perform the quality control analysis. So today we can just use the data and perform the data integration using FastMNN and Liger. First, we can use FastMNN. MNN stands for Mutual Nearest Neighbors. This is another method that has been reported to integrate single-cell RNA sequencing data from different batches or sequencing data from different technologies. FastMNN identifies cells that have mutually similar expression profile between different experimental batches or replicates, then corrects batch effect by matching mutual nearest neighbors. So first we need to normalize the data. We can just use the AML data after the quality control. Let's run the normalization data function. Then find the variable features. So for the FastMNN analysis, we don't need to run the step of scale data. We can just straight go to run FastMNN function. And we know we have 3 datasets merged into the AML Surat object. They are AML1, AML2 and AML3. And we can differentiate them from the origin ident in the metadata. This is very similar to the standard Surat data integration method. We need to split the object into an object list. But the step of integration by run FastMNN function is much quicker than the standard Surat integration method. Let's run FastMNN. You can see it's very quick, and also it uses the 2000 features from all 3 datasets. So after FastMNN we can run UMAP. Next, we find the neighbors. Then find the clusters. Now we are ready to visualize the cell clusters. We can group the cells by their origin ident to see the batch effect, and also we can group the cells by the Surratt cell clusters. Let's run the dimp node function, and we can zoom in. You can see on the left hand side are the cell clusters labeled by their sample names. You can see now after the analysis, the cells were integrated very well among 3 datasets. On the right hand side we have 27 cell clusters. So now I showed you how to perform data integration using another method called FastMNN. You can see it is a very quick method to do the analysis. We can set the FastMNN analysis as plot 3. Later we can compare with the other method called Liger. Now I can show you the Liger function to integrate single-cell RNA sequencing dataset. Once again, we are going to use the merged AML Surratt object from 3 single-cell RNA sequencing dataset. So the first 2 steps are the same as other analysis. We need to normalize the data, then find the variable features. So for the Liger analysis, we also need to scale the data, and we split the merged AML data again into a data list by their origin ident. To scale the data, because the matrix factorization in the Liger function requires positive values, so we don't need to center the data, so we can set to center as false for the data scaling. So let's run data scaling. You can see down here, they scale the data separately from AML1, AML2, and AML3. So after the data scaling, we can run the key functions for the Liger package. So if you want to understand the details of the Liger package, you can go to the online tutorial for the Liger package. They have very clear steps for the data analysis. You can see the first stage to pre-processing and normalize the data. We already did this for our long data set. The next one will be joint matrix factorization. They have details for each parameter in the optimized ALS function. You can see the key value. According to the online tutorial, a key value range from 20 to 30 works very well for most dataset. Because this analysis is an unsupervised analysis, so there's no correct key value you can set. If you want, you can try different key value to see your final cell clusters. Also, you can set the lambda function. The default value is 5, and they recommend use 5 for most analysis. So let's run the function optimize ALS in Surratt. We can set the key value as 20 and the lambda as 5. Then split the data again using their origin ident. Let's run. The Liger function is a little bit slower than the Harmony and the FastMNN. This step takes 2 to 3 minutes for our dataset. Okay, it is done. You can see it takes around 3.4 minutes to run the optimized ALS function. So next we can go to stage 3 to run the contour normalization and the joint clustering to identify the cell clusters. So we are going to use the default value for the contour normalization function in our analysis. You can have a look at all the parameters. What is the default value for this function and what is the best value for you to change it to analyze your dataset. So let's run this function for the stage 3 analysis. Now we can run the UMap. Then use the dimplot function and group the cells by their origin ident to see data integration using Liger function. Let's zoom in. You can see the cell clusters. Again, you can see cells from different samples are integrated very well using the Liger function. So let's set the Liger function plot as plot 4. Then we can visualize them with the standard workflow, FastMNN and the Liger analysis together. We can zoom in. You can see the batch effect from the standard workflow analysis. Both method, FastMNN and the Liger function, integrated the cells very well from different sequencing dataset. So, for data integration in Surat, I introduced the standard Surat integration method and also the harmony method in my previous video tutorial. In this video, I showed you how to use the FastMNN method and the Liger method. You can see all the methods worked very well for the dataset we analyzed. So now it's your choice to choose one as your favorite data integration method. For me, I quite like the harmony and also the standard Surat integration method. So I'm going to stop for today's video tutorial. I hope my video can help your data analysis. Please subscribe to my channel if you like my videos. Thank you and see you next time.